solved the first and highest resolution structure of low density lipoprotein in native state at human body temperature. So before joining IIIT Delhi, he worked at a personal genomics company in Malaysia for a year, followed by Genome Institute of Singapore for more than 8 years, and has published some fundamental research findings. And he is primarily working on epigenome and gene regulation, and has also applied these findings to other branches such as cancer and stem cell study using data sciences and genome. With this brief introduction, I would like to invite Dr. Vibhor Kumar to take over the session and present his work.
Thank you, Jaspreet. So let me, so you can see my presentation?
Yes, we can see.
So okay, so I hope only my presentation is visible, or also this Zoom window is also visible on the side?
No, only the presentation, but it is not in full screen mode.
Okay, I see. Okay, thanks.
Yes, now it is.
Okay, so today I'm going to present the opportunities and challenges of a single cell search engine for healthcare applications. So a few years ago, it was kind of I and Professor Devarka, who is we used to walk and talk that how it can be implemented, and we used to discuss, and then we came up with this idea, and that how much it will affect, it will help the society and the community. So we started working together, and then later on I pulled for single cell epigenome also. So starting with some introduction about single cell profiles. So you might be already aware of single cell expression profiling. Nowadays, single cell epigenome profiling is also being done widely. Then now coming up is single cell proteome, is the protein expression of single cell. And then mutation profiling for every single cell, and single cell lipidomics also. On the right-hand side, you can see spatial single cell transcriptomics and proteomics, which is on the horizon. And so there have been a few manuscripts where researchers have shown profiling of almost 10,000 genes from every single cell with their spatial locations. Right? So it is moving ahead. But even without spatial information, just the single cell expression itself, the analysis and utilization of it, it has a lot of scope. So as more and more people are coming up with new ideas. So as I told you, that there are still many challenges and scope in single cell data sciences. So Lahneman et al. Published a very interesting study, review in genome biology, where they talked about 11 challenges, grand challenges in single cell data sciences, out of which 5 are very relevant to single cell expression profiles, epigenome profiles. So I have mentioned here 5. So basically, first thing is handling the sparsity of single cell profiles. So as we know that the material that we'll get from every single cell will be so less that there's a high chance of dropouts. That's why it is very sparse. Moreover, when you are using the same resource for a large number of cells, like the read counts, you don't get so many read counts from large number of cells. So, and second challenge is how to develop some statistical frameworks for discovering complex differential patterns. So what do you mean by complex differential patterns? So basically, in a cell, if in a community of cells, there is heterogeneity, right? And so there is a differential pattern among groups of cells. The way the angle you look, you will find different kinds of differential patterns among different groups of cells. You may discover new groups of cells, right? So if you just look at from the perspective of oxidative stress, then you will find that, okay, these cells are oxidative, or these cells are stem cell and non-stem cell, whether they belong to any lineage, doesn't matter. So the defining such kind of flexible statistical framework is the second challenge. Third is mapping single cells to a reference atlas. So currently there are huge data sets being published, but how a common man can compare his own data of single cell expression profile, huge data sets, and generalizing trajectory inference, like by this we mean that if you have developing embryo and you have got cells from developing embryos, and you have combined embryos from multiple time points, then you want to know which cells actually came first, which came later. Or if you have a growing tumor, then you have to find which cells came first and later. So this kind of trajectory inference, like what is the trajectory of the growth or the differentiation of the cell? So that is one of the challenges. And of course, a bigger challenge is that a lot of groups are producing single cell expression and epigenome profiles, and they have their own protocols and own types of measurement. Sometimes they use different species model. How to combine all of them to make a senseful insight? Because if you have batch effect and you can't just use expression data from two different labs together, because it's not easy to actually use conventional tools, visualization tools like tsne and umap to actually see them together. So I'll be talking about one of the challenges is mapping single cells to a reference atlas, right? One of the 5 challenges. So this is just with a motivation to benefit from published use cell atlases, single cell expression and epigenome profiles, to reduce the redundancy, introducing new data sets, right? So how we can do that? So suppose, if we want to study some cell line models, right? So we just need to say that, okay, I will look in the web. If we have published cell lines, then I can use that. But that is another thing. But if there are certain cells coming from in vivo samples, right? It is not easy to match your own data to in vivo samples, because most of them, they are not labeled properly. So, and then finding lineage and poisoning of cancer cells, because often cancer cells show intermediate phenotypes of multiple cell types. And of course, annotating new cells. And from there, annotating new cells comes with a great application in liquid biopsy, that annotating circulating tumor cells. So basically, this is a new kind of liquid biopsy coming up nowadays. It's expensive right now, but in future it's going to be very helpful. Is that when cancer develops, it sheds cells some cells are shedded into the blood, and those cells can be actually purified, and their profile expression and epigenome profiling can be done. But how to find that they are actually cancerous, or from which organ they are originating, right? You have application of mapping to reference atlas. And of course, understanding diseases. Nowadays, these huge consortiums of single cells are moving towards profiling expression profile from disease samples, single cell expression profile from disease samples. And of course, validating rarity of cell states. So basically, if you found a new cell state and you want to claim it, so that it would be very worthy to actually check it on the to compare it with the existing reference atlas. But if there is a huge batch effect, it might be very hard. So there, this challenge of mapping single cells to reference atlas comes in, right? And in 2018, we published probably world's first single cell search engine, and so it was published in Duplicity Research. And after that, we were followed by other groups. But let's go to the challenges. So I talked about the utility, and now challenges. The challenge is that the size of reference atlas is so huge. I can tell you frankly that there are more than 10 million single cell expression profiles freely available, more than 1 million single cell epigenome profiles available, but they also have batch effect because there are different protocols to actually profile expression, like UMI, SmartSeq, MarsSeq, etc. And there is a day-to-day variation in expression profile, even from a single person. Even if a single person is doing single cell expression profiling, one day you take batches from two different days, you may see a batch effect. Okay. And then there is cell-specific biases also. In the same data from the same lab, if you have different cells, maybe they have different noise level, different dropout rate, right? Because of stochasticity in distribution of the read counts. And of course, as I told you, very high dropout rate causes sparseness of data. So among all these problems, you have to solve this problem of making search engine for single cells. So we started with handling first the bigger challenge is how to use a large reference atlas and match the user expression profile to our reference atlas. And so basically we use hashing approach. So we use LSH, locality sensitive hashing. The main idea is that we take a random projection of the cell and we put based on the binarized bucket binarized, we come up with some we come up with buckets of cells, groups of cells, right? When the user puts his expression profile, single cell expression profile, they are also projected according to the same random projection planes, and then we get a binary code and we compare those binary codes. So accuracy of hashing based approach was 60 to 65%. So, and that was not enough. It increased the speed, but so we had to add one more layer of statistical filtering. So basically, what we did is that once we have hash codes for for the reference as well as for the query, so we calculate the distances, but then we compute the p-value using those distances with the help of a null model. So basically, we have smartly made null model, so that our null model is composed of 1,000 false queries, right? And then we compare the distances of those 1,000 false query cells with our reference expression profile. And then whenever users, so if we have, suppose a user's query single cell, we've got this distance from our reference expression profiles. Then we compare those distances with the distances of the same reference expression profile from the null model. Then what happens? We got a p-value, and when we introduce this p-value, the accuracy increased by 20 to 25%, and then it helps a lot. So the benefit of this is that it could handle the cell-specific biases, like if a reference cell, for a reference cell, suppose a particular distance is the maximum, right? Even if you give a similar cell, because the data is so sparse and noisy for that reference expression cell, single cell, then you can't get more than certain distance, right? So for every reference cell, we have a reference a distribution of distances, right? And using the null model, and that helped in actually filtering out the artifacts in the distances, right? So this gives a cell specific, this gives a control over the cell specific biases. And it helped. So basically, we also implemented it on GPU. So you can see that our GPU graphical processor unit-based search was much faster than CPU. And then we also tested it with different dropout rate. So we had this, we already had like a sparse single cell expression profile of SCT116 cells, but we further created dropout, simulated more dropout in those cells. And we found that what accuracy we get till like at different rates of dropout. And then and then so you can see here, okay, so at different for different cell types dropout rate. And then we published it in nucleic acid research, and after that we were followed by several other groups, and they published it in Nature Communication, Nature Methods, PMC Genomics, so in variety of journals. And so we are happy that a trend was started because of our initiative, and actually Lahaneman et al. Have actually acknowledged our publication. But this is not that what we made was a preliminary version, right? So we have, our vision was to actually make something that is useful for clinicians and researchers. So my group further investigated on this topic, and so we are in the process of making we made it a next generation cell search engine, which adapted transfer learning and self-learning. It has self-learning with like adaptive AI approach. So basically, we use deep learning. So if you use simple autoencoder, right? So you give it a data single cell expression profile from multiple batches, multiple sources, and you train the autoencoder, and then you get this reduced dimension representation of the single cells, and you further look at using t-SNE based embedding. You could say that, okay, if you get a training data, I am able to actually group cells according to types, right? Of course, here also there is some problem, because if you have a major cluster and you have a minor cluster, minor group of cells, there also autoencoder clubs the minor cells, minor cell or rare cell population with the major cell population. So it's not perfect, right? So you can see here, but if you take same cell type data, but from different batch, from the unknown, from what we have written here, unseen batch effect. So from a different lab whose data was not seen by the autoencoder, right? And you do a simple transfer learning, not adapting, simple transfer learning. So you train it on your own data set, right? And then you take data from same cell type, but from other unseen batch effect. And you can see that all the cell types mix, right? Typical autoencoder that will do. But we have modified this. So we have modified in the way how you use on autoencoder. And so we use autoencoder in a semi-supervised way. And you can see here, even training data and data with unseen batch effect. so you can see here, it groups well. And so like here, you have green and yellow. So these are both same cell types, actually. But yellow is with unseen batch effect. And when I color according to the cell types, you can see here, they're all according to the cell types, no mixing. So this is, and what is so interesting is that our algorithm actually allows to learn from the query itself. So once it groups them properly, it actually further adapts from using the query pattern. So it has self-learning. So basically, basic idea is that now once you make this a small search engine, it grows by itself, right? It grows itself, and it needs very little control. But of course, we would be definitely checking it every 15 days or 1 month. So this is coming up. And but this approach is our approach is novel, and it can be generalized to multiple different kinds of search engines, which we are currently exploring with 3 different kinds of deep learning approaches: autoencoder, GANs, and LSTM. So, and with different multiple applications, we are trying that, but it's not so straightforward and easy to implement. So now coming to a new kind of search engine, that is for the single cell epigenome profiles, because as I told you that there are already more than 1 million single cell epigenome profiles available. So the trend for search engine for single cell epigenome profile has not yet started. So yes. So let's give a brief overview of single cell epigenome profiles. So basically, you can see here from a cell, you can have different kinds of epigenome profile. So you can have chromatin interaction, histone modification, DNA methylation, chromatin accessibility. Okay. So what are the benefits and promises of single cell expression profile over RNA? So first of all, from clinical samples, epigenome profiles are more preserved. If you take circulating tumor cells or old clinical samples, the RNA has destroyed, like it has been broken down, and so their epigenome signal is still intact, which can be used. And then you can have a better inference about direct stimuli, because you can see here expression profiling just captures the profile of 1% of the genome, right? That's the gene bodies, but here you can explore 99% or rest 99% of the genome also, because external stimuli works through the whole genome, right? Through enhancers and depressors. And of course, you can make inference about different kinds of regulatory elements. And as I told you, it will give you, because since you can see the effect of external stimuli better, you can actually infer the information about the potency, the effect of external stimuli and the potency of cell in a better way. Okay. So we made this SAP search. It's probably the first search engine for single cell epigenome. So just to give an overview of how epigenome signal look like. So basically you have this whole, there are different cells, and in a single cell, you can see here, you get the read counts of open chromatin, and we convert this, we call peaks, based on the read counts, and these peaks, instead of genes, we have peaks, becomes features, and the read counts becomes the feature scores. So we get a read count matrix. And the problem is that if you have data from different labs, right? So they have different peak lists. So first that we do is we have to bring them to a common platform, right? So and then single cell expression profiles are these read count matrices are much larger and they're much sparse. And of course, the batch effect is also there, because you can have open chromatin profiling using single cell ATAC-seq, SCI ATAC-seq, single cell DNA-seq, single cell MNA-seq. So there are also different protocols, just like RNA-seq. So to start tackling the challenges, so what we do, we first calculate gene enrichment score using the peak read counts. Okay? And So basically, gene enrichment score, before calculating gene enrichment score, we actually highlight enhancers by normalizing the read counts with offer that peak with the global accessibility score. So we get more cell type specificity of the peak, and then we assume it as enhancer, and then we take proximal genes and we use Fisher's test to calculate gene enrichment score. So, and then to match it to the reference expression profile or to the reference open chromatin profile, we just don't use correlation, and LSH and hashing will not work here. That's out of scope. So we use very simple rules of how enhancers or how epigenome works, right? So they work through genes. So what do we say that, okay, we have query, we take top 1,000 enriched genes of the query, okay? And then we look at those, that query, if you have top 1,000 enriched genes, we look at their expression profile in the reference, our reference single cell. So our reference single cell, we have like 3.5 million single cell expression profiles. So for those cells, we find what is the median value of these 1,000 genes in that query, right? And the one that has highest, we say that, okay, most likely it is the matching. But even there, we have cell-to-cell variability adaptation. And it's the same thing. So basically, for the reference single cell epigenome profile, we have already, so instead of gene expression profile, we use gene enrichment scores. And we see the top gene enrichment, median gene enrichment score of the top 1,000 genes of the query. So like this, we are able to actually reduce the batch effect that would have come due to correlation or due to hashing, because hashing doesn't promise you the batch effect reduction. Okay? So benefit of that is that they're not able to. Of course we got good epigenome to single cell epigenome to single cell epigenome match, as you see here. So here you see the percentage of cells where you get the right hit in the top five results. Okay? So this is human single cell epigenome to human single cell epigenome matching. And then for human expression profiling, like so matching to human expression profiles, also it gives quite satisfactory result. But our approach also help to match query human cells, a single cell attack profile, to reference expression profile from mouse. So we can do a cross-species search. So you can find a matching mouse models for the single cell open chromatin profile from the cell, from humans. So we could use it for annotating some unknown cells from mouse thymus, which was published by Kosanovic et al. So what we found is that matching expression profile gave a lot of white blood cells and T cells, and we know that thymus is actually enriched in T cells, which are type of white blood cells. Okay, so now this is very trivial, right? So catching the cell type, annotating cell is kind of trivial for known cell types, but how to use it for more complex problems? So we use it for understanding the multipotency in cancer cell line. So for example, if you see K562 cells, it has been shown that it can differentiate to macrophages and dendritic cells, of course, and erythroid. So the first lineage that is differentiated to is erythroid lineage by default, right? So SAP search actually gives the match scores of query cells with the reference epigenome file. So we use that for hierarchical clustering, and we found 2 clusters of K562. So if we just use top 1,000 genes, right, of the query cells, for finding the matching expression or epigenome profile. The top hit that we get, of course, is erythroid, that is well known, erythroid-like erythroid precursors. So cluster 2, for cluster 2 cells, we got only erythroid. For cluster 1, we also got other types. But these embryoid body cells, they are not properly annotated. So we said, okay, we take top 2000 genes, which also could be consisting of poised genes, right? So we found that cluster 1 cells also get hits from macrophages, dendritic cells. The Langerhans are also kind of dendritic cells, and some bone marrow mononuclear cells. Whereas cluster 2 always gave match to erythroid. Okay. So when we added the p-values of match of these different cell types, what do you find? That macrophages have higher, like more significant p-values for cluster 1. This is for cluster 1 cell. So this shows that cluster 1 cells are more poised to differentiate towards macrophages than actually erythroid-like, right? Similarly, they could be a dendritic cell. So cluster 1 cells have same potency towards dendritic cell as towards erythroid. And then we looked at the frequency of genes. So basically, we get gene enrichment score for different cell types, right? For different cells. So we say that, okay, which genes are appearing in top 50. So, and then we calculate the frequency of genes to be enriched in top 50. Okay. And when we compare the frequencies of genes appearing in top 50 for cluster 1 and cluster 2, we found that certain genes, which are known to be marker of dendritic cells, they have higher frequency of gene enrichment score in cluster 1 than in cluster 2. Okay? So it shows that there are some genes which are activated or poised more in cluster 1 cell in respect to cluster 2. That actually keeps the cell poised, K560 is poised, cell poised for differentiating toward the lineage. So this explains that it's the heterogeneity in the cells, which could be responsible for multipotency of K562 cells. So now coming to revisiting these grand challenges. So as I told you that there is one more topic, that is integration of single cell data from different experiments and groups together is also a kind of challenge that has been mentioned by Lenham et al. And so when you do the literature search, you will find a lot of embedding methods proposed for single cell expression profiles. Very, very rarely you will find it for single cell epigenome profiles. Okay, so reason being is that single cell expression profiles, you have genes as common features, right? And it is quite old field, but in single cell epigenome profile, it's not yet fully explored. So so since SAP Search has an approach that allows matching across species and batch, so we said, okay, we can actually co-embed different queries. So basically, what we did is that we have a standalone version of this tool. In the standalone version, we entered actually multiple. User can actually enter multiple read counts. So those read counts may have their own peak list. So you get data from read count matrices from different groups with your own peak list. You can enter them all in the standalone version of SAP Search. And then what we found that SAP Search is actually able to get a lower dimensional representation of the cell. We can say the embedding, such that same cell types cluster together, irrespective of the batch or the species. You can see here that we have human hematopoietic stem cells sitting together with human mouse hematopoietic stem cells, the results from SAP Search. And then we use other methods which were proposed for just scale was proposed for single cell epigenome profile. It just didn't work. And then other methods which were proposed for single cell expression profiles, they just completely fail for co-embedding of single cell epigenome profiles. Even if no matter how you extract the features, so they have their own way of latent variable extraction, but we tried all that, they all failed. It means, so what does this say? That there could be a lot of technical knowledge, ways, but this very basic, simple mechanism of how enhancers regulate genes, how epigenome analysis is done, right, leads to such kind of results, which is much, much stronger than all these fancy latent feature extraction methods. And this is a very simple example where we took B and T cells. You can see here B cells from different species are clustering together. T cells from different species are clustering together. And we could actually validate it using DV scan. So basically on the embedding results, you could actually further do partial clustering and calculate the purity. But we didn't stop here. We just moved on and we say that, okay, how it can help further, this co-embedding, how it can help further. So basically first we took data of a single cell ataxic profile from normal and a leukemia patients. So this leukemia patient had this multiple phenotype acute leukemia. And so when we when we put this normal cells, blood cells in the SAP search, the result from SAP search, yes, anybody has any query? So results from SAP search shows that the normal healthy individuals, the cells, they show the match of the distribution of the matching expression profiles as expected. So there are T cells which is known to dominate, be a dominating cell type in blood, and then you have monocytes. It is according to the proportion that is expected. But when we use the cells from multiple phenotype acute leukemia to patient cells, and we found a completely distorted this proportion. So we found a lot of dendritic cells matching to, so we found a lot of dendritic cells matching to the MPAL cells. Okay, of course, T cells were there. So we explored further. It could be because of cell selection during profiling. It might not reflect the true proportion. But for PBMC, for healthy cells, it shows it reflects the true proportion that is expected. So further, what we did, okay, we took a single cell ataxic profile of differentiating early progenitors, hematopoietic stem cells, like hematopoietic stem cells, and then MPP, okay, CMP, CLP. So I have shown here how they differentiate to different lineages on the right side. We took their single cell ataxic profile, and then we also took a single cell ataxic profile of B cells and T cells, okay? And then and these are all from different groups, okay? And then I co-embed using SAP search. We co-embedded that with a normal PBMC cell, normal patient, normal human, like healthy individuals PBMC cell. What do you see? That healthy individual PBMC cells, blood cells, they're clustering together with B cells and T cells, and the MPL, this multiple phenotype acute leukemia cells from patient 1 and patient 2, they were more towards undifferentiated. So a version of hematopoietic cells, and they were overlapping. Few of them actually overlap completely with undifferentiated progenitors of hematopoietic cells, and they all it's multiple phenotypes. So they were actually overlapping with different lineages, right? So they were overlapping with CLP cells, myeloid progenitors, as well as leukemia progenitors. And even when we compared the cells from two different patients, we found very less overlap among leukemia cells from two different patients. Even though they were drawn, or they were closer to to similar progenitors, but they are not exactly overlapping. So there was patient-to-patient heterogeneity. At the same time, they had a dedifferentiated state, which was similar to progenitors. So that explains the plasticity of blood leukemia cells. Okay, but I mean, but we don't stop here. So what do we expect in near future? How this kind of approach will help us for healthcare? So as I already showed you that we already have an adaptive search engine which has self-learning capacity. We are trying to make it more lightweight and lab tailored, so that you can use it on Google Colab, just like that student can use it in Google. So that we are in a process of making that. And then second challenge is what is coming up is that soon different groups across the world will be producing single cell profile from diseased. We already have some from diabetes, but more and more is coming. So how to actually help clinician in matching their single cell profiles with the diseased? That can be done to some extent, but the problem will be there could be some same cell type, undiseased and diseased, right? So we have to match to the exact diseased ones. And second, second one is that there could be some unseen state, disease or stress state of the cell, right? Which would be claimed as rare, but actually there could be disease, right? So it has to be smart to that extent that even if it has not seen a disease state, it should be able to say that this looks like disease. So we are trying to build up, trying to add that feature. Of course, it has to go through some markers and other focused feature-based approach. And then the main vision of this is actually to help clinicians to drive the search for suitable drug for a precision medicine. So basically, if clinician's disease expression profile matches with cells from patients from a different corner of the world, so clinicians can actually connect with each other, right? And then they can actually discuss with each other. Yes, looks like okay, this drug was suitable. Let's try this, right? So that's a new platform that is coming up on our single cell search engine. So I think researchers. So the conclusion is that we are pioneer in building a single cell search engines for expression profiles and epigenome profiles, and there is possibility to search and co-embed irrespective of batch effect, species, cell-to-cell variability, and there's possibility to have a smart search engine which grows itself like a child and become very smart adult search engine. So we have actually started the child to learn and to train it, and let's see how far it goes. And we can actually fine-tune it according to the lab's own requirement. And then still, as I told you, there are many things to be done in future. There's a long journey ahead, but it's worth it. So finally, I would like to acknowledge, first of all, my friend, Dr. Devarga Sen Gupta, with whom we started this one. And recently, Kedar from SDA from Denmark, he has also joined in, providing some novel data to be analyzed and reported. And our PhD student, Nitesh Divyanshu, Divyanshu was a master's student. He has published like 3 papers with me. And now he's a PhD student in Switzerland. And Arvind, also a PhD student in Switzerland now. Shreya Misra and Smriti Chawla. I am open for questions.